Hi, this week we are starting chapter 12, enzymes. We are finally done with the macronutrients, and now we're going to kind of dive into a little bit more of a nitty-gritty of food science. After enzymes, we'll be covering micro components of food. The enzymes kind of fall under proteins, but there's no extra like dietary things with them. So we'll do micronutrients and phytochemicals. So that's a little bit of nutrition there with those, and then after phytochemicals, we will be getting into uh, more ingredients and how you use different ingredients as substitutions for food, so food analogs, and use ingredients to produce desired characteristics in food, etc. So, get started with this. Okay, so what is an enzyme? If you've had chemistry, you should be pretty familiar with this, and this little graph on this slide should look familiar. So enzymes are proteins that speed up or start a chemical reaction without being changed by it. And because enzymes are not changed by the chemical reaction, they can be used over and over again. And because of that, many enzymes are found in pretty low concentrations because they can fulfill their function by just being constantly used. Um, an enzyme is just a special version of a catalyst. So a catalyst is defined as a substance that speeds up a reaction between substances without being affected by the reaction. So the only difference between an enzyme and a catalyst is that an enzyme is a protein catalyst. Okay, a catalyst that is a protein. Not all catalysts are proteins. Um, so almost all chemical reactions in the body use enzymes, and thousands of enzymes can be found in a single cell because we need enzymes for everything, almost everything that our body is doing. Enzymes are reaction specific. Okay, so each enzyme only catalyzes a specific chemical reaction. And that's why we have to have so many different ones because there's lots of different chemical reactions going on in our cells. Um, Now, a lot of times, although technically the reactions could happen without the enzyme, in practicality they wouldn't because too much energy would be required. Um, and so I'm show you this here. This is kind of what enzymes do is they lower the amount of energy that is necessary for a reaction to proceed. So this particular reaction requires a level of energy that is this high for it to occur without an enzyme. But when you add an enzyme to it, now the level of energy that is required for that reaction to happen drops. Okay. And sometimes quite significantly. So, for example, for digestion of foods, without enzymes, so much heat would be required in our bodies to digest the food that it would actually need to be boiling inside our stomachs to digest the foods that we eat, and they still wouldn't digest in an as efficient manner as they do naturally with the enzymes that we have in our stomach and our digestive system. Okay, and so we would not be able to live without enzymes. Now, why this is important for food scientists is that enzymes they can degrade food, so they can be something that we sometimes fight, and we want to inactivate the enzymes to stop food degradation. But also, enzymes can be used by food scientists to make specific food products. So we can use them beneficially, and we sometimes need to know about them so that we can inactivate them and prevent them from harming our food products. So there are a couple different models that are used to explain how enzymes work. The first is the lock and key model, and I'm going to modify my explanation of this model to make it be a combination lock. Um, so in this model, the lock is the substance that is being changed. Okay. So we need that lock to be opened. Okay, and that's how we're going to change it. Okay. And the substance that's being changed can also be called the substrate. Okay, so that's an important term because I'll be using that a lot. So the substance that's being changed, our reactant, is called the substrate. Then the dial in our combination lock, or a keyhole if you're using a regular lock analogy, um, is the location that the substrate attaches to the enzyme. So without the combination, the lock cannot be opened without a great deal of difficulty and effort. And this is time that most people don't have, right? But if we have the combination, we can easily open it. But we must enter the combination in the correct order in the correct way, right? And if you're talking about a key, you must have the right key that is shaped the right way, and it must enter in the correct way. All right, so this is kind of a diagram of the lock and key model. If this substrate is not turned the opposite way, if you rotate it, it's not going to go in, right? And so that's what I mean by that. It must be positioned a specific way. So in this model, and so this, the keyhole or the combination, the dial, represents the active site of the enzyme. Okay. So in this model, the lock represents the substance being changed or the substrate, and the keyhole or dial represents the location that the substrate attaches to the enzyme. Okay, and this is a visual model that is more lock and key and not like combination lock. Okay, and you see how the pieces fit in perfectly there. Okay, so the second type of model that is used to explain how enzymes works, and so by models, this is just like ways of explaining it. It's not exactly, and different scientists believe that one model or the other explains how things work better. So the second model is the induced fit model, and with this model, they suggest that the enzyme actually will adjust its shape in response to the substrate. And then that initiates the reaction. So you see the enzyme here, it's not exactly the same shape as the substrate, but then the substrate inserts itself, and the enzyme actually adjusts its shape to have a fit. And initiates that reaction, and then you get the two products. Okay, so all enzymatic reactions have three parts. Okay, three steps, I should say. So you have the substrate and the enzyme separate. Okay, and so that is demonstrated here in this first step of each of these diagrams. Okay, and then you have the enzyme substrate complex. Okay, those are demonstrated in the second step. So that is when the substrate and the enzyme are bound together. Now this particular set of models introduces a third step, which in this set of three, it would be included here. It's just dividing this one into two steps. This one says, okay, so once that reaction occurs, it's not immediately separated. And so you actually also have an enzyme product complex. Okay. And then finally the products are released. And so you have separately product and enzyme, or products and enzyme. Okay. So another term to know in regards to enzymes is what coenzymes are. And so coenzymes are additional non-protein substances that must be present for some enzymatic reactions to occur. Not all enzymatic reactions require coenzymes, but some do. Now notice this red here, I added that because coenzymes are non-protein. And so that is how you can distinguish them from enzymes, okay? They work with the enzymes, the protein, to catalyze the reaction, okay? Um, so there's three mechanisms in which coenzymes may work. So depending on the enzyme-coenzyme combination, they use one of these mechanisms, okay? So different coenzymes work through different mechanisms. The first is that the coenzyme attaches to the enzyme so that the combined shape reacts with the substrate. And that's what's being shown here in this diagram. Okay, so you have the enzyme and the coenzyme actually sits in the enzyme before the substrate binds to it. The second mechanism in which coenzymes may work is that instead of attaching to the enzyme, they attach to the substrate. And so once they're attached to the substrate, now the substrate actually fits in the enzyme, whereas it didn't before. And then the third potential mechanism is that the coenzyme accepts the atom or molecular group that is broken off the substrate by the enzyme. So if we look at this, for example, so let's say that the enzyme produced these two products, but these two products react very easily with each other, okay? And so what could happen is this enzyme produces these two products, but as soon as it releases them, they rebond and we're back to the same original substrate. And so what this third mechanism is saying is that the coenzyme will come in and bond to this product, for example, preventing these two products from rebonding together and just going back to being the original substrate. Okay, and last thing is that coenzymes, they can be a single ion, like calcium, even an electron, or it can be a larger molecule. The only caveat is that they are non-protein. Okay, so enzyme nomenclature. Nomenclature simply means naming system. Okay, so you could say enzyme naming system. How do we name enzymes? And there's a few ways that they can be named. Quite often we see that they are named for the substance they catalyze. This is the easiest way when you look at an enzyme name to know what it's actually doing. So for example, lactase is the enzyme that catalyzes the breakdown of lactose. Okay, and so you'll see part of the substance part of the name of the substance that they catalyze, so lact and lactose, is in the name, and then you add ase. Okay, so when you see ase at the end of the name of a substance, you know that's an enzyme. Okay, so lipase would be a substance that breaks down lipids. Protease, substance that breaks down proteins, an enzyme that breaks down proteins. Carbohydrase is an enzyme that breaks down carbohydrates. Now these three are very general terms. You don't have enzymes that are just called this. This is lipase, right? There usually are specific names, but this is a general category of these enzymes. So enzymes that break down lipids are lipases. Enzymes that break down proteins are proteases, etc. Okay. Now we can also see enzymes that are named for the reaction that they catalyze. So catalase, this one is very generically named, but it is named for the reaction, and it's named catalase because it catalyzes hydrogen peroxide into water and oxygen. Then the third way that they can be named is just because they had another common name nomenclature before this system of naming the enzymes was created. So renin, Renin, the properties of renin have been known for a very long time, okay, for centuries. And so it was named well before we had a specific way that we named enzymes. And in fact, probably didn't know renin was an enzyme, or what an enzyme was when it first got its name. But renin is a protease used to curdle milk. Okay. Invertase just has another common name. Its regular nomenclature name is sucrase because it breaks down sucrose. But sucrase produces invert sugar, produces separately glucose and fructose. And so it is also called invertase because it produces invert sugar. So now we'll go through factors that affect enzyme activity. And the first one is water availability. So water is sometimes a reactant, okay? When we make macromolecules, so when we make lipids and carbohydrates and proteins, a water is released, okay? So you take glycerol and a fatty acid, and a water is taken from the juncture where that bond is formed. When a glucose and galactose bond to form lactose, water is removed from the spot where that bond is formed. When peptides bond, water is released from where that peptide bond was formed. So conversely, to break down these larger molecules, we need to add a water molecule. Or sorry, yeah, to break down these larger molecules, we need to add a water molecule. So here is triglyceride, and you have just a generic lipase that is breaking it down. You need to have water added in that reaction, and you remove one of the fatty acids. And so you notice here you have, okay, two hydrogens, a carbon, and 2 oxygens, okay? Well, here we have 2 hydrogens and the carbon, and then we have 1, 2, 3 oxygens and the products from where that bond was, as well as 2 more hydrogens, 1 here and 1 here. And so that's where the water went. One of the hydrogens went here, and one of the oxygens and the other hydrogen went here. Okay. And so that's just one set of examples where, and the same thing goes for carbohydrates and peptides being broken down. That same kind of thing is going to happen. So that's just one set of examples where enzymatic reactions require water as a reactant. Now, water can sometimes also be a solvent, or is also a solvent. And so water as a solvent, being the liquid that everything is suspended in, that gets the substrate to the enzyme so the enzyme can do its job. Without water, there's no way for the substrate to, number one, reach the enzyme, and number two, be positioned correctly. And so drying is one way that we can generally stop enzymatic reactions from occurring in food products. A second factor affecting enzyme activity is solution concentration. And so that's simply the greater the concentration of the substrate, the greater the rate of reaction is going to be. And that's kind of universal with any reaction. The greater concentration of your reactants, the greater the rate of the reaction. And this exists up to the saturation point of the enzyme, and I'll show you what that means, okay? So here we have a low concentration, and it's just going to take longer for each substrate particle to reach an enzyme molecule and be positioned in exactly the right location and that reaction to take place. The more molecules of the substrate that you have, the more likely, as things are just moving around in solution, that it's going to hit one of the enzymes and hit it in the right place, and the um, reaction take place. And so here you have, two of them happening at this time. But there does come a point where the amount of substrate that you have exceeds what rate these enzymes can even take it up and perform the reaction and then release the product and then take up another. It just exceeds the rate that can happen. And so that is the point of saturation, where the enzymes are always going to be working at that moment. And all of the enzymes in solution, have a substrate bound to it and are working. And then you just have a lot of excess substrate that is just waiting around for the next time, an enzyme is free. And so at that point, the reaction rate is constant. It doesn't matter how much more you add of the substrate, it's not going to happen any faster. The third factor affecting enzyme activity is enzyme denaturation. And within that, there are several ways that enzymes can be denatured. The first one is heat. And we know that reaction rate increases as temperature increases. Okay, we've talked about that before, whether you're in chemistry or earlier on in food sciences. As the temperature increases, the reactions will increase. Unless, however, an enzyme is involved, because if the heat is so high that the enzyme becomes denatured, then now that enzyme won't be active, right? And so that is what this graph here is demonstrating, right? It's just as the temperature increases, we just keep seeing an increase in reaction rate. But at a certain point, at this random temperature here, this representing something for some enzyme, that enzyme becomes denatured. And at that time, the reaction rate will plummet as more and more of those enzymes become more damaged. Okay. So most animal enzymes, so enzymes within animal bodies, denature above about 40 degrees Celsius. Catalase is a common enzyme that we deal with, and that is inactive at 60 to 65 degrees Celsius. And so it just depends on what enzyme you are trying to inactivate or trying not to inactivate, and knowing what that denaturation temperature is and staying within that range or above that range if you're trying to inactivate it. So continuing on with heat, so as I kind of suggested, sometimes we do want to stop enzyme activity, and heating can be a way that we do it. And one really good example is blanching, where we usually do this to vegetables, fruits or vegetables, and you briefly plunge that food in boiling water to stop enzyme activity. And then it's typically followed by submerging the food in ice water to stop the cooking action, because it's not trying to cook the food, you're just trying to preserve it, because usually after blanching, you'll then, a lot of times it's done before you freeze it, and you're trying to keep them green and keep the flavors fresh during its frozen storage of those vegetables. Okay. Meat tenderizers is another example. So you add meat tenderizer, you usually will let it set for a little bit to let that meat tenderizer take effect. But as soon as you cook that meat tenderizer is inactive. It's not going to get any more tender after you cook it. Okay. Second way that enzymes can be denatured is through acids or bases. And so each enzyme has an optimum pH range for reactions, a certain range of pH where it's going to perform its best. So for example, pepsin, the enzyme in our stomachs that helps break down protein, has a pH of pH optimum range of 1.5 to 1.7, which is very low. And that makes sense because our stomachs have hydrochloric acid in them. Okay. Amylase, which breaks down starch, has an optimum pH range of about 7. So this isn't in our stomach, it's further down in the intestines, and its optimum pH range is close to 7. Rennin, which is used for cheese making, has an optimum pH of 5.5 to 6. And polyphenol oxidase, so polyphenol oxidase is the enzyme that turns apples brown, has an optimum pH range of 5 to 7. So that's when it is most active at turning apples brown. Okay. Now, enzymes will, so let's back up. So milk pH is about 6 when we're talking about rennin. So milk pH is really close to the range where rennin is not very active. And in fact, it can start to denature at above pH 6. And so for rennin to work, acid needs to be added to milk to get it more within rennin's optimal pH range. Okay. Now polyphenol oxidase, which we want to inactivate if we want to keep our apple, like if you're selling sliced apples, for example, if you want to keep that apple from turning brown, you want to have this enzyme in a range where it's not as active. And so you'll add acid to inactivate, probably not denature, but have it be less active and save your apples from turning quite as brown by adding something like lemon juice. Okay. Now to denature this particular enzyme, it has to be below pH 2.0, which is really low. And so you'd have to add a lot of so much lemon juice that you'd be really affecting the flavor of the apple. And so you typically won't be able to really fully inactivate the polyphenol oxidase, but you can get it into a range where it is not as happy, not as optimal, and not working as well. Okay, and then a third way to affect enzymes through denaturation is through electrolytes. And so electrolytes is just a fancy name for the positive or negative charged ions in solution. Higher concentrations of electrolytes can either hinder or promote enzyme activity depending on what they are and what the enzyme is. We often use salt, which is a combination of two electrolytes. When it goes into solution, it's going to separate into sodium ions and chloride ions. And so salt is commonly used to stop spoilage enzymes and preserve products. Iron, calcium, and potassium work as coenzymes for some enzymes, and so those can actually promote enzyme activity for better or worse. And then you would not add this to food, but mercury and lead are ions that will stop enzyme activity. And you're not going to add those to foods because they're going to stop enzyme activity in the human body too, which is why mercury and lead are so poisonous in the human body, and you do not consume them. Okay, so the next factor affecting enzyme activity, and the last one is enzyme inhibitors. And so enzyme inhibitors are any substance that will prevent the enzyme substrate enzyme substrate complex from forming. Um, when the shape of a substrate or enzyme is changed, they are unable to fit together correctly, and the reaction is going to be stopped or slowed. And so that's what these inhibitors will do. They're going to bind with and change the shape of either the substrate or the enzyme. Okay, and these inhibitors are used by biological systems, so they're naturally in our bodies to prevent microbiological enzyme activity. So, for example, in eggs, the inhibitor conalbumin binds with the iron in egg whites to stop bacteria and yeast that need free iron to grow. And so it is a natural antimicrobial that is part of the egg white that helps protect the egg and the developing chick inside it from microbial growth. Okay. Another biological example is a lot of inhibitors are used for metabolic regulation, so regulating our metabolism. And an example that you'll often see is that the products will often act as inhibitors until the concentration is low enough that more production is needed. And so here's an example here. You have this substrate 1 reacts with the first enzyme, produces a product, which reacts with another enzyme, which produces a different product, which reacts with a third enzyme, which produces this third product. Well, the level of this third product, if there is above a certain concentration of it, it's going to react with this first enzyme and prevent this A to B reaction from happening. And this is called a negative feedback loop. And it's very common in biological reactions. And the main goal is that you don't want to produce too much of this. It helps produce only what is needed. And that is the end of this week's lecture. It's a pretty short one, even though it was more than half of the chapter. So yay for this week and only a little over 30-minute lecture. And we'll see you next week when we cover the rest of enzymes.